Hi, Anjay. Why don't you tell us about your role at Eurofins and what you do for the company?
Sure. Yeah. So I am a subject matter expert in probiotics. I have a background in food microbiology. My official title is Senior Technical Manager, um, but I also run the genomics department in our laboratory. I'm based in Madison, Wisconsin, and I've been in the microbiology field for about 15 years now.
Beautiful. So, this episode, we're going to focus on whole genome sequencing. So, why don't you start by telling us what whole genome sequencing is?
Sure. Yeah. So whole genome sequencing is the determination of the nucleotides in anything. So humans, bacteria, animals, think back to Jurassic Park and finding out the DNA in a mosquito's genes or something like that. So it's the determination of characteristics of something as well as the identity of things using the DNA in a sample.
Yeah, perfect. I mean, it's a great topic, and I'm a genetics major, so this is kind of where I'm going to geek out a little bit.
Sure.
So, why don't you tell us about how the technology with whole genome sequencing has evolved over time from sort of like the whole genome project back in the 90s to where we are now?
So it originally started back in the 70s, and with Sanger sequencing, super slow, relatively expensive. Um, in the 90s, the whole genome project came to be, and that's when we started looking at second generation sequencing. It became a little bit faster to sequence things in parallel. And so, but at the same time, it was still extremely expensive, cost prohibitive. We're talking millions of dollars per genome. In 2003, the Human Genome Project was finished, and they completed the genome. And shortly after that, then next generation sequencing was developed. There are a few companies that looked at this new technology, and essentially what it did was make things a lot faster and way cheaper. In 2007, things were still tens of thousands of dollars to sequence. Fast forward 10 years, and we're looking at maybe $1,000 in 2015, and a few different types of technologies then continued to make things more cost effective for scenarios like my laboratory, which needs rapid, high throughput, fast and cheap testing.
Yeah. So you mentioned next generation sequencing. So what is next generation sequencing, and how's the technology been used to aid preventing foodborne illnesses?
Sure. Yeah. So next generation sequencing, there's really two main types: long read and short read. Long read sequencing uses a particular technology that is able to look at the sequence of nucleotides that can be up to 100,000 base pairs long. Super cool. It uses nanopores, and it's really good at identifying structures. So, like looking at the identity of a particular organism. Then there's short read sequencing, which looks at smaller fragments of and but has a great deal of precision, a great deal of accuracy, and is used in a wide variety of disciplines, and is really good for looking at like certain functional characteristics of genes. Those are the two main types, and then as the technology has advanced, as these companies have continued to make advancements in the kit chemistries they're using and advancements in the actual instrumentation, things have become much more precise, and much more streamlined. Like I said, 10 years ago, we were looking at a cost prohibitive amount to be able to use whole genome sequencing in like a third party lab setting. It's just no one would be willing to pay, you know, $10,000 to figure out an ID when they could get a biochemical ID for less than 100. Today we can effectively sequence an entire genome and look at it, for less than $1,000. Where and 25 years ago, this was
Yeah.
$10 million. And so it's crazy just how fast the technology has advanced since the turn of the 21st century, and I think it's still continuing to evolve, and I think things are going to continue to become cooler and cooler, because it is something that really excites me. My background's in probiotics, but the genomics field is just so fascinating and how fast it's evolving over time, especially in the food safety space. So, talking more specifically about how whole genome sequencing can help in that area, there's two main aspects to it. There's the kind of the uh, cause and effect sort of relationship of how it can be used. So, like after an outbreak, we have a salmonella outbreak in leafy greens. We can use whole genome sequencing to characterize that particular strain of salmonella, and then look at the food supply, look at the environment, look at the illnesses and the people that got sick and the strains associated with that, and try to make linkages using whole genome sequencing. This wasn't as easy in the past. We were using things like pulse field gel electrophoresis to establish relationships. And what whole genome sequencing has done is really made things a lot. They've provided a lot more resolution, and that's really helped tie, to try to find where the sources of these outbreaks are coming from. Is it coming from an ingredient? Is it coming from environmental contamination that's being brought into a plant by XYZ? So that, there's that part of it, but then there's also kind of the preventative aspect and the use of whole genome sequencing. So environmental monitoring, and not only, to identify that the source of contamination is Listeria monocytogenes, but to say it's Listeria monocytogenes strain XYZ, we know this is a transient strain or this is a house strain in this plant, and then they can address it accordingly. This is a type of information that we didn't necessarily have a few decades ago that is really powerful and can be used in a food safety setting, a food manufacturing, a supplement manufacturing setting to aid in preventing anything from going out that could cause illness.
Yeah, it's a super powerful tool. And I think also it can be used by manufacturers to clear them of wrongdoing. So if someone has a salmonella illness and you can't prove that, you can say, well, it's a different strain than perhaps salmonella that's in our environment, right? And so you can use it also, in the inverse. So with infant formula specifically, Chronobacter is the big problem. Salmonella as well, but really those two are the big microorganisms. So, what can you explain to our audience a little bit about what Chronobacter zakizaki is?
Sure. Yeah. And just tying that into the infant formula topic too, this is something that's extremely important in the aspect that um, these strain identities are imperative in these sorts of investigations, thinking back to everything that happened in 2021 and 2022 with the recalls and everything like that. Whole genome sequencing was used, and it was determined that there were no definitive linkages between what was in the infant formula, what was found in the plant, what was found in the illnesses, and that was a critical piece of information in that whole piece. So, tying that back into what Chronobacter is and why we care. And it has become, much more prevalent since all that happened back, a few years ago. But this organism has been around for millions of years. It was originally discovered in the 30s. I think it was first isolated in 1950. It was originally classified as part of the Enterobacter family. Um, but the reason we've determined it to be a public health concern is that it's very tolerant to dry conditions, so it can survive days in a desiccated environment. So that poses problems for anything that is dry. So powdered infant formula is a great example of a matrix that could potentially support Chronobacter survival. Not only that, but Chronobacter, it is an opportunistic pathogen. It's ubiquitous, but it's the age group with which it can particularly cause some really bad health effects that is of major concern, and that's because it's 0 to 2 months, really. So newborn infants, premature infants, infants with a reduced immune system, and it can be particularly dangerous to that target population. So obviously, super super sensitive population that even a few cases is a big deal. And we typically see, the FDA has seen 2 to 4 cases documented a year. But even still, this is the sort of thing that we don't want to see any. And the problem with Chronobacter being ubiquitous is not only is there potential sources of contamination from ingredients or things like that, but one of the biggest risks is actually, the preparation of the formula at home and ensuring that the process is being followed and the correct procedures and the correct, the surfaces are clean, because you can bring in Chronobacter into an environment on your hands, on your feet. I mean, that's usually how it comes into plants and things like that. So, um, that's why it's a particular concern, and it has really become a lot more mainstream since the shortage post-pandemic.
Yeah. I think that's a key point, is that, yes, it can come into the plant, but it's very much in the homes, and the home is not going to be a GMP environment. It's not going to be, super
Right.
sanitary
They're not
necessarily.
swabbing.
No, we're not swabbing the house for
Right.
sure. So, you can only control so much as a manufacturer.
Right.
Once it gets to the consumer, it's kind of up to them to keep everything as clean as possible.
Mhm.
So, what is the FDA strategy to help prevent Chronobacter zakizaki illness due to consumption of powdered infant formula? They obviously aren't going to be in the homes of parents who are making the formula. So, what is their strategy to help?
Yeah, well, part of it is a public awareness
Hmm.
campaign, and ensuring that um, people and mothers and parents that are using powdered infant formula understand the risks associated with Chronobacter illness when they have these susceptible populations, and understanding just basic, you know, microbiological sanitation prevention practices. That's just one, obviously, small aspect to it. The FDA is kind of looking at it as a whole, and I'm not going to speak. I can't speak for the FDA. I'm not
Sure,
affiliated with the FDA.
sure.
I just know, what they're looking at, and really they want to look at it as a risk-based approach, like many things.
Yeah,
So establishing models at the manufacturing level, and the production level, distribution level, to determine, where the pain points are, and then the effective strategies to mitigate those particular areas. And it's learning. A lot of it is just learning, what trends are associated with Chronobacter contamination.
yeah,
Where is it coming from? How do the strains survive? And using microbiological techniques and genomics techniques to aid in that. Not only looking at the identity of the strain, but also the functional characteristics of certain genes that may not be associated with just Chronobacter sakazaki, but there may be like a E. coli
yep,
0157, STX gene or something like that. So getting a better understanding of Chronobacter as an opportunistic pathogen, similar to the amount of literature out there for Salmonella listeria, is one big part of it.
yep.
And then also increasing their oversight, or their, not oversight, but their
They're monitoring.
yeah, yeah.
Yep,
So, increasing their awareness to look at these facilities and ensure that the best practices are being performed, and educating the manufacturers as
yep,
well as to the best practices. And in the end, everyone needs to be held accountable for
yep,
ensuring that our food is safe, and that's what we do as third party
yeah.
testing labs too. And then, doing the best we can, to educate the consumer on the best ways to use these
Yeah, it,
products.
it's an ongoing battle for sure. So, where does whole genome sequencing fit in with the FDA's monitoring program?
Sure. Yeah, they called out whole genome sequencing specifically in their guidance document as the tool to use to effectively manage, mitigate, and control Chronobacter sakazakii. If the laboratory or receives a presumptive positive, the FDA expects that that positive will be taken to whole genome sequencing to establish at a strain level what this organism looks like. You can then do a lot of things with that. You can compare it to the NCBI database, see if there are any linkages already out there.
Yep.
You can compare it internally. Perhaps a company already has an internal database of strains that they've sequenced
Yep.
that they're able to use then to help help prevent it, help mitigate the contamination event in order to keep production going.
Okay, so how does NACMF get involved with the FDA's initiative here?
Sure. Yeah. I love NACMF. They're a great group of scientists and
Or maybe we need to explain what
epidemiologists.
NACMF is.
Yeah, sure. It's the National Advisory Committee for the Microbiological Criteria of Foods.
Yep.
And they are advisory committee as part of the USDA Food Safety and Inspection Services. So they are currently in a 2-year cycle,
Mhm.
and they have 2 charges related to the outbreak and subsequent investigations, and really to help the FDA strengthen their regulatory oversight of Chronobacter and infant formula.
Yep.
So the first one is an FSIS charge that's related to genomics. That's a particular group of scientists that are looking at some more advanced techniques for helping determine risk associated with these organisms, things like targeted metagenomics, deep sequencing, and multi-locus sequence typing, and trying to get a better picture of exactly the potential risks associated with each particular strain. And maybe it's not necessarily strain dependent.
Yep.
It could be a particular gene of interest. So they've really taken a deep dive into that, the
Yep.
genomics group. And then there's another charge that's coming from the FDA that is related to the Chronobacter in infant formula, and that's more of a public health initiative. Looking at ways to aid in assisting the public in understanding Chronobacter illness, what it means, the risks associated with Chronobacter and infants, and ensuring that they have the best picture to give, or the best guidance to give the FDA to support their initiative.
Perfect. So last but not least, where does Eurofins fit in with this? What can we do to help support the FDA initiative with whole genome sequencing? What is our lab doing? Feel free to go into that detail.
Yeah, sure. So my lab specifically in Madison, I'm part of the microbiology group. We do long read sequencing
Yep.
using Oxford Nanopore, and we are able to characterize to the strain level. So fast, accurate, precise genomic characterization of strains. And we can do that for pathogens
Yep.
as well as other things such as spoilage organisms, probiotic strain identification. It really is a powerful tool for bacterial ID or any microbiological ID, virus, things like that as well. We also do a lot of metagenomic sequencing, and this can help in certain situations like spoilage investigations and looking at probiotic blends. But when you're talking about, pathogens and determining pathogen identification, a lot of the stuff is happening at the confirmation level.
Sure.
So a laboratory receives a presumptive Salmonella, Listeria, Chronobacter, and then the next part of that step in the test is to take that and learn more about what's going on with that presumptive hit. Is it a true presumptive one? And if it is, what is it? And that's where whole genome sequencing can really assist with just one of many, many things it can do.
So just one last question. I know I said the last one was the last
No,
question,
please.
but this is another question. Do you think the biochemical methods of identification are going to go away in other industries in favor of whole genome sequencing. So I know for infant formula for sure, it's the FDA's pointing that way, which makes sense. It's a very sensitive population, right? It's important to get it right. Do you think things like 16S sequencing, Vitek, API, etc., is that going to go away in favor of whole genome sequencing with that becoming cheaper as the years go on? Do you think other industries will also turn to whole genome sequencing?
I so I think there will probably always be a place for 16S.
Hmm.
I do think that the advantages of genomic characterization and whole genome sequencing will eventually make biochemical identification obsolete.
Sure.
Once it gets to the point where it's equivalent from price and turnaround time perspective,
Yeah.
and then it is significantly better with all the other criteria associated with the databases, the quality of the identification, et cetera.
Yeah.
Uh, I do think there will be a paradigm shift, but
Yeah.
things like 16S and MLST, I think there will be obviously places for those in certain aspects of the production environment
Sure.
when it needs to be even faster and
Yeah.
cheaper.
Yeah. Well, thanks so much for coming and talking to us about whole genome sequencing, Anjay. It's been a great discussion.
Thanks for having me.
And yeah, it's been great. Thanks so much.
Appreciate it.